Inside every single living thing, there's this hidden universe of constant activity. It's a cycle of growing, copying itself, and dividing that, well, it underpins life itself. But this process isn't just simple. It's an incredible biological challenge that every cell has to solve with absolute precision. So let's dive into this elegant and, yeah, pretty complex story, the cell cycle. So let's kick things off with the central engineering problem the cell is up against. I mean, think about it. It's a packaging puzzle of truly epic proportions. How on earth does it manage to cram this enormous library of genetic information into a nucleus so tiny it's almost impossible to imagine? And believe me, that number is no exaggeration. If you could actually take the DNA from just one of your cells and stretch it all the way out, it would be about 2 meters long. That's taller than most people. And all of that has to get packed into the cell's microscopic nucleus. So, how in the world does the cell pull this off? Well, the solution is nothing short of a masterpiece of biological engineering. It's a multi-level packaging system that takes what would be a hopelessly tangled mess of thread and turns it into a perfectly manageable, super condensed structure. And this shows you exactly how it's done, step by step. It's this beautiful hierarchy of coiling. First, that long DNA strand wraps around these little protein spools called histones, which makes it look kind of like beads on a string. Then that string of beads coils up into an even thicker fiber. From there, that fiber gets arranged into a whole series of dense loops held in place by a protein scaffold. And finally, the whole looped thing gets compacted even more to create the dense chromosome we see when a cell divides. I mean, the final result is an incredible 10,000-fold reduction in length. Just amazing. The real heroes of this first step are these histone proteins. They form a core, an octamer to be specific, that acts just like a spool for thread. And the secret sauce here? It's all about basic chemistry. See, DNA has a negatively charged backbone, and these histone proteins are packed with positively charged amino acids. Opposite charges attract, right? This creates a really strong ionic bond that lets the DNA wrap tightly and securely around that histone core. And what's really cool is that not all of this packaged DNA is treated the same way. The cell can actually control which parts of its genetic blueprint are accessible. You've got euchromatin, that's the beads on a string form, which is open for business, ready for its genes to be read. And then you have heterochromatin, which is super condensed, tightly packed, and basically silent. It's kind of like the difference between having a book open on your desk, ready to read, versus having it packed away in a locked box in the attic. Okay, so once the DNA is perfectly packaged up into these neat little chromosomes, the cell is ready for the main event, division. Now, this isn't just some chaotic, messy split. Oh no, it's more like a meticulously choreographed dance called mitosis, and its whole purpose is to make sure each new daughter cell gets a complete and perfect set of instructions. This whole process, which gives us 2 identical daughter cells, happens in 4 main stages. First up is prophase, where the chromosomes get all condensed and ready to go. Next, in metaphase, they all line up perfectly right in the middle of the cell. Then comes anaphase, where the copies are pulled apart to opposite sides. And finally, we have telophase, where 2 new nuclei form around the chromosomes, finishing the job. Having a little trouble remembering that order? Hey, no worries, I gotcha. Here's a great little memory trick. PMAT. Just think of it as prepare for division in prophase, they meet in the middle for metaphase, they move apart in anaphase, and then you make 2 new cells in telophase. Prepare, meet, apart, 2. It's a super simple way to keep those stages straight in your head. Now, after the nucleus is divided, the rest of the cell, the cytoplasm, has to split too. This part is called cytokinesis, and it actually happens differently depending on the cell. Animal cells form what's called a cleavage furrow that basically pinches the cell in two, kind of like pulling the string drawn on a bag. But plant cells, they've got a rigid cell wall, so they can't do that. Instead, they build a new wall right down the middle, called a cell plate. It's like building a partition to divide one big room into 2. Okay, as you can imagine, this whole intricate process of packaging and dividing is way too important to just be left to chance. It's all tightly regulated by a really complex molecular control system. Think of it like a team of protein supervisors and gatekeepers that are constantly telling the cell when it's okay to move on to the next step. Right at the heart of this whole control system are enzymes called cyclin-dependent kinases, or CDKs for short. The easiest way to think about them is as the engines of the cell cycle. They're kinases, which is just a fancy way of saying they add phosphate groups to other proteins. This acts like a switch, turning proteins on or off to push the cycle forward. But these engines can't just start up on their own. And that's where their partners, the cyclins, come into play. If CDKs are the engines, then you can think of cyclins as the keys that turn them on. The levels of these cyclins rise and fall throughout the cycle. And it's only when the right key, a specific cyclin, is present that it can turn on its specific engine, the CDK. For example, to get the green light to start copying its DNA, the cell needs cyclin D and cyclin E. Later on, to actually enter mitosis, it's cyclin B that acts as the key for the main division engine, CDK1. And to make sure everything is perfect, the cell has these critical checkpoints. They're basically quality control stops, kind of like a pre-flight check before an airplane takes off. The G1 checkpoint asks, hey, are we big enough? Do we have enough nutrients? Is the DNA undamaged? Then the G2 checkpoint double checks that all the DNA has been copied completely and without any major errors. And finally, the M checkpoint makes absolutely sure that every single chromosome is properly attached to the spindle before they get pulled apart. No mistakes allowed. So who's in charge of these critical safety checks? Well, one of the most famous proteins in all of biology, P53. It's often called the guardian of the genome for a very good reason. P53 is constantly monitoring the DNA. If it spots any damage, it slams on the brakes, pausing the whole cell cycle so repairs can be made. And if the damage is just too bad to fix, P53 makes the ultimate call. It triggers the cell's self-destruct sequence. This is exactly why P53 is so crucial, and why, when it's mutated, which it is in over half of all human cancers, a major safety system is just gone. And that brings us to the cell's ultimate fail-safe mechanism. When a cell gets damaged beyond repair, or it's infected, or maybe it's just old and not needed anymore, it doesn't just fall apart. Instead, it undergoes a clean, orderly, programmed self-destruction. The process is called apoptosis. Now, this is totally different from another kind of cell death called necrosis, which is super messy and usually caused by an injury. Apoptosis is like a controlled implosion. The cell shrinks down, breaks up into these neat little packages, and gets cleaned up by other cells without causing any inflammation. Necrosis, on the other hand, is like an uncontrolled explosion. The cell swells up, bursts, and spills all its guts out, which triggers a big inflammatory response. Think of it as a planned, careful demolition versus a sudden, messy building collapse. The agents carrying out this controlled demolition are a family of enzymes called caspases. You can think of them as the cell's own executioners. They hang around in an inactive state, but once they get the signal, they activate and set off this deadly cascade, systematically chopping up all the essential proteins and dismantling the cell from the inside out. So where does the order to die come from? Well, it can come from two different directions. The extrinsic pathway gets triggered by an external signal. Think of it like another cell sending a self-destruct text message that binds to a receptor on the cell's surface. This kicks off a chain reaction that starts with an initiator called caspase 8. The intrinsic pathway, however, is triggered by internal stress, like if there's severe DNA damage. That signal comes from inside the cell, from its own mitochondria, which activates a different initiator, caspase 9. But in the end, both paths lead to the exact same place, activating those executioner caspases that get the job done. So we have this unbelievably intricate system of division, quality control, and even self-destruction. But what happens when that system breaks down? What happens when a cell just starts ignoring all the stop signs and flat out refuses to die? Well, that, right there, is the fundamental basis of cancer. It's a disease of the cell cycle, and it's caused by mutations in two critical types of genes. First, you have proto-oncogenes. These are the gas pedal, and they normally tell the cell to grow and divide. When they get mutated into oncogenes, that gas pedal gets stuck to the floor. Then you have tumor suppressor genes, like our friend P53. These are the brakes. When they're lost or mutated, the cell loses its ability to stop. So cancer is often the disastrous result of having both a stuck accelerator and a complete brake failure. And how exactly does that gas pedal get stuck? Well, it can happen in a few different ways. Sometimes, a single tiny change in the DNA, a point mutation, can jam a protein like RAS permanently in the on position. Other times, the cell can accidentally make way too many copies of the gas pedal gene, which just overwhelms the system. Or you can have something called a chromosomal translocation, where the gene gets moved to a new spot, putting it under the control of a hyperactive switch that's always on. And this really brings us to a final, powerful thought.
Our cells have checkpoints, they have repair machinery, they have a self-destruct sequence, all these safeguards designed to prevent exactly this kind of uncontrolled growth. The fact that cancer can happen at all is a sobering testament to the incredibly delicate balance that's required for complex, multicellular life to even exist. It's a disease that's born from the very same processes that allow us to grow, to heal, and ultimately to thrive.